Bowers is online. All right, so Sharp, David, Hodges, Sipple's online, Eric, Midkiff, got you. Paulie, Rice is here. Cedric, Richardson is online. Massey, Newton, Burdette is online. Hudson, Felder, Tudor is here. Asbury's online. Bowers is online. Hemmings, Redmond is online. Reed, Michaela's here. Taylor's online. Van Cross. Gotcha. And Cedric is online now. Got less than a minute left. Any questions while we wait to officially start? Anybody do anything really fun over the spring break? Nobody? That's okay. Me either. I did a whole lot of nothing, and it was glorious. Nice to relax for a bit. All right, let's see. Once we get started, there we go. It's 8 o'clock. I'm still gonna wait till 8:01. Make sure nobody else shows up. But once we get started, we're gonna finish up here on chapter 8, and that'll be it for the day. So we might have a short day, unless of course you have lab. There's really no announcements. Just the normal reminders. Read the announcements that I post. If you haven't retaken the second exam yet, I would highly recommend it. You have nothing to lose, even if you somehow get a lower grade. We'll just keep your higher grade. I think only 5 or 6 people retook the first exam. It's up to you. I will say, though, because we do 8-week semesters, so one of my classes just finished, and I got the inevitable emails that I know I'm going to get for this class. So if you care about your grade, work on it now. So, like at the end of the semester, be like, is there anything I can do about my grade? If you didn't retake the exams and you didn't do the study guides and things like that, then obviously you don't really care enough about your grade. So do it if you need to. And as always, if you have any ideas for independent work, extra credit, please let me know, because I would like to make that easier for you, because like I said, we're halfway there. So you should have about 50 points by now. And if you don't, it's a good time to work on it. And let me know if you have some ideas of things you would like to do other than write papers. So any questions about anything? Okay, let's jump back into it. Back into chapter 8, which is all about cellular reproduction. We actually just finished talking about meiosis, but way back when, a week ago, which is perfect timing, because we're going to go through a really quick review where we compare mitosis and meiosis, and then we're going to talk about genetic variation and talk about what happens when meiosis goes wrong. And that's a perfect segue into the next chapter, which we'll hopefully start on Wednesday, which is all about genetics, which is what we're doing in lab this week. So here we go. Again, we're comparing mitosis and meiosis. So before I even jump into this, I'll remind you how it's going to be on the exam. Mostly the questions for mitosis are going to be like, in what stage did this happen? In which stage does that happen, right? All about the chromosomes. For meiosis, it's going to be a lot of how is meiosis different than mitosis. So here we go. For both chromosomes, or for both meiosis and mitosis, the chromosomes duplicate only once, right? So that all is the same. Chromosomes duplicate in interphase, specifically S phase of interphase. As a matter of fact, I wish it said that. Let me fix this here. S phase of interphase, because you do need to know that for the exam. Actually, that'll be the first word for attendance. For those of you who are online, or if you guys here want the extra credit, the first word for attendance will be S phase. or I guess the first term. Anyway, the chromosomes only duplicate once, whether we're talking about mitosis or meiosis. However, the number of times it divides is different, right? So in mitosis, it only divides once. So you duplicate yourself, right? So double the number and then cut it in half, right? So if you double something and cut it in half, you're back to the original number. And that's what mitosis is, right? One division, and that produces two diploid cells. Meiosis is different, right? So you have the one duplication, then you cut it in half, so you're back to the original number, then you cut it in half again, which means you're at half the numbers, and that gives you 4 haploid cells. So there's a lot of information on here that would be on the exam, right? So for mitosis, there's 1 division. For meiosis, there's 2 divisions. For mitosis, you end up with 2 cells. For meiosis, you end up with 4 cells. For mitosis, you end up with diploid cells, and for meiosis, you end up with haploid cells. Any questions about this? All right, and again, just because it's been so long since we've been together, I'll remind you, mitosis is used for asexual reproduction. So you're probably doing mitosis right now as you sit here. And meiosis is for sexual reproduction. Meiosis is what we use to produce sperm and eggs. And then obviously those sperm and eggs come together in fertilization, producing a diploid zygote. So any questions about this slide? Okay. The events that are unique to meiosis 1, and this will be on the exam, or excuse me, the events that are unique to meiosis occur in meiosis 1. And another way of saying that, and this might be written like this on the exam, is meiosis 2 is essentially the same as mitosis, right? The way it's happening. Because in meiosis 1, remember the big change, the big difference, it's not on this slide, is that we are separating homologous chromosomes in meiosis 1. Because remember, you have 2 pairs of each of the 23 chromosomes, unless there's something wrong. You know, like chromosome number 17, you have 2 of them, one from your mom, one from your dad. Chromosome number 12, you have 2 of them, one from your mom, one from your dad. And in meiosis 1, they all get together, right? And those are the things that get separated. And then once those are separated, then finally in meiosis 2, that's when we separate the sister chromatids, just like we do in mitosis. And again, in meiosis, we end up eventually with these haploid cells, right? So they don't have the full amount of chromosomes, which makes sense because, again, in meiosis, we're producing sperm and eggs. So we want to have half the genetic information. So any questions about this slide? Okay. Here's a picture of everything we said. You know, again, just comparing meiosis to mitosis. Mitosis, again, you duplicate once, cut it in half once, and you're back to the original number. That's mitosis. In meiosis, you duplicate once, you cut it in half twice, and you end up with 4 haploid cells. And of course, too, this stuff that'll be really important later. You can see like these red chromosomes have a little bit of blue on them, and these blue chromosomes have a little bit of red on them. So to remind you, that is crossing over, right? The chromosomes kind of swap chunks sometimes in prophase 1. So your mom chromosomes get a little bit of dad on them, and your dad chromosomes get a little bit of mom on them. They swap chunks. So any questions about that slide? Again, this is just a review, which is why I'm kind of going through it quickly. All right. This brings us to the next main bullet point. This is an important bullet point, the origins of genetic variation. So the next chapter is going to be about genetics. And this week's lab is about genetics. So obviously this is an important piece of the puzzle here because we're just introducing it now. We're going to talk about it a lot. And then once we get done talking about genetics, we're going to talk about evolution. And you couldn't have evolution without genetic variation. So this is a very important bullet point, not just for the exam, but for understanding what's coming up in the semester. So as you should know, and as you need to know for the exam, as I've already said, the offspring of sexual reproduction are genetically different from the parents and one another, as you should know, right? You know that. You are not a clone of your mom. You're not a clone of your dad, even if you're really similar to one or the other. You're not a clone. You are genetically unique. Also, if you have siblings, you're genetically unique to your siblings, right? So how does meiosis produce this genetic variation? And we've already talked a little bit about it, so some of this will be a review. Not this first one, though. Here we go. First thing we're going to talk about is independent assortment of chromosomes. You definitely need to know what independent assortment is. So we'll go through this slowly. You need to know what independent assortment is. So when the homologous chromosomes line up in meiosis, in that first metaphase, the way they line up is completely by chance. Meaning, I've shown you the picture 1.5 million times, but, we'll try to picture it, and I'll show you a picture, an actual picture later. You've got the homologous chromosomes lined up like this, right? And imagine that 23 times, right? Because you have 23 pairs of chromosomes. And it's not like what I'm saying here. Independent assortment means it's not like all mom on one side and all dad on the other, right? It does not happen that way. The way it happens is completely random. So I guess, yes, technically, maybe there could be a chance where they were all mom on one side and all dad on the other, but it's not very likely, right? They just randomly line up. So again, if you have siblings, even though full siblings, even though you share the same genetic information like from your mom and your dad, I don't know, again, let's just say randomly, let's just call that chromosome number 1. You may have gotten your mom's chromosome number 1. Actually, I won't get into it yet. Let's just say independent assortment, again, they just line up randomly, right? It's not all mom chromosomes on one side and all dad on the other. It's line up randomly. So for a species that has over 2 pairs of chromosomes, every pair orients independently of all others. So again, just a technical way of saying the way they line up is completely random. And then, of course, we've got to get math involved to make things a little bit more difficult. This is a number I'm going to put a question mark. I might ask you this. I'm not big on numbers, but this is one where I'm not sure yet. I have to think about it, but I might ask you this on the exam. So since we have 23 pairs of chromosomes, again, imagine trying to draw it out. That would be a hell of an independent work project. If I were to tell you to draw out all the different possibilities of, the mom chromosomes on one side and dad on the other. The reason that would be such a hard thing to do is because there's 8 million combinations. So when you produce sperm or egg, there's 8 million possible combinations of which chromosomes they get. Because again, it gets cut in half, right? So when you produce sperm or produce egg, and I'm going to randomly say, chromosome number 20. When you give a sperm or an egg, it's either going to be your mom's chromosome in there or your dad's. It's completely by chance. We don't know which. And if there's 23 pairs of chromosomes, that means there's 8 million possible combinations. Another way of saying that is, statistically, if you were to look at 8 million if you were to look at 8 million of your sperm or egg, then statistically they should all have a different orientation of chromosomes, right? And that's just your egg or sperm. Then consider the fact that we're talking about fertilization, right? So you produce a gamete, your partner produces the gamete, and then they fertilize. So that means there's 64 trillion possible combinations. Another way of saying that is if you had a sexual partner and you had 64 trillion kids with them, statistically, they should all be unique, right? Their combination of chromosomes would be unique. 64 trillion of them. And remember, that doesn't even include crossing over, which I'm going to talk about here in a second, but we've already talked about that in the previous lecture, right? So yes, you might, the mom chromosome, the dad chromosome, but again, your mom chromosomes aren't all mom chromosomes anymore because they crossed over, right? So you have that mom chromosome that has a chunk of dad on it, or a dad chromosome that has a chunk of mom on it. So yeah, it's really crazy when you think about these numbers. 64 trillion possible combinations. So if your parents, assuming they had sex to have you and not like IVF or something like that, if your parents didn't have sex at the exact time they had it, in the exact way they had it, you wouldn't be you. There would be some other sperm getting together with the egg, and you would not be you. You might be very similar, but you would not be you. You are one in a 64 trillion. Right? These numbers are, to me, very amazing. You think about how unique unique you are. Again, your parents could have sex or have 64 trillion offspring. And statistically, they should all be unique. And again, that's just independent assortment. That doesn't even include crossing over. So anyway, any questions about what independent assortment is? All right. So here's just showing you the different possibilities. Of course, this is an organism that has 2 pairs of chromosomes, right? And we have 23. So look how, even with just 2 possibilities, even with 2 pairs of chromosomes. In this case, you have all the mom chromosomes on one side and all the dad on the other. In this case, they're flip-flopped. And I'm just going to say blue is dad. Why not? So you got a dad chromosome and a mom chromosome, and then a mom chromosome and a dad chromosome. So you can see these different combinations. And again, that is with just 2 pairs of chromosomes. Again, imagine that with 23 pairs of chromosomes, which is what we have. Staggering numbers. Anyway, any questions about that slide? Okay, like I said, those 64 trillion combinations or possibilities don't even include crossing over. So like we already said, crossing over is the exchange of corresponding segments between homologous chromosomes. So again, as a reminder, you have 23 pairs of chromosomes. For each chromosome number, you have 1 from your mom, 1 from your dad. They get together in prophase 1. They're right next to each other, and sometimes they literally swap chunks, right? Sometimes it's a small chunk. Sometimes it's a big chunk. Sometimes it's multiple chunks. Sometimes it doesn't happen at all. So again, you got those 64 trillion combinations, but then you also have crossing over. So there's not even always mom chromosomes and dad chromosomes, right? You got mom chromosomes with a chunk of dad and vice versa. And of course, this next slide I'm going to show you is just going to be a visualization of what I've just reminded you of. So there's no new information on this slide, but we're talking about it again. So you need to know what crossing over is. And of course, that one's pretty simple, right? It's kind of in the name, crossing over. The chromosomes cross over. Independent assortment, in a sense, is more important academically because, one, it's a little bit harder to remember. And we're going to talk about it again in the next chapter. So independent assortment, we're not just like talking about it once and we're done with it. This is going to come back to haunt us, so to speak. So make sure you understand what independent assortment is. And yes, crossing over, but honestly, academically, independent assortment is a little bit more important. So any questions about this side? Any questions about crossing over before I show you a slide? It gives you a visualization of crossing over. The next word for attendance, I'm going to circle it. I won't even say it. So if you're watching online or if you're attending online, make sure you open your eyes. Because it's a Monday after spring break. And if I were in your shoes, I would be listening to this lecture with my eyes closed. But I don't want you to fall asleep. Because also, if I was in your shoes, this would make me fall asleep. So open your eyes, look at the word that I circled, and that's the next word for attendance. Now, let's move over to the next slide and see a visualization of what we're talking about of crossing over. There it is. You see those chromosomes, those homologous chromosomes right next to each other. Got a pair from your mom, a pair from your dad. Well, when I say a pair, it's because they're technically sister chromatids. They cross over. So here you go. Now you have a dad chromosome with mom chunks on it. And then, of course, you have the mom chromosome with dad chunks on it. And then they separate in meiosis 1, and then separate again in meiosis 2, and that's what you're left with, right? So just looking at this one chromosome pair, you have these different options, right? So you produce 4 gametes. One's completely dad. One's mostly dad with mom tips on it. One's mom with dad tips on it. And one's completely mom. So that's crossing over. And again, add that to the 64 trillion possibilities regarding your gametes and your spouse's, your mate's gametes coming together. Right? These numbers are pretty astronomical. Anyway, any questions about this? All right. What happens when meiosis goes wrong? We already talked about what happens when mitosis goes wrong. And just to remind you, you need to know that. Of course, it's not here. That was a smaller bullet point of the second main bullet point. But remember, when mitosis goes wrong, basically cancer, in a sense, right? When the cell cycle is out of whack and you just keep reproducing cells, that's cancer. Let's talk about what happens when meiosis goes wrong. And these are all really great independent work topics. So we're going to scratch the surface of these things. I'm not going to tell you much about them. I'm not going to test you on most of them. So if you're interested, you can look them up for independent work and dive a little deeper. All right, what happens with errors in meiosis? It can result in genetic abnormalities ranging from mild to severe to fatal. So as a matter of fact, a lot of times when there's, I don't even want to say a miscarriage, like when the zygote doesn't even form or keep going, a lot of times it's because of this, problems with meiosis. So let's talk about it. How accidents occur. Basically, you need to know this word, non-disjunction. That's what we call it when something goes wrong in meiosis, non-disjunction. And it's kind of in the name. It's one of those words on the exam that should be easy because it kind of describes what happens. Non-disjunction. So they're just not separating properly. The chromosome pairs fail to separate at anaphase. Of course, the fact that it says at anaphase, that's a given. That's more like a reminder of what you should already know, which is that chromosomes separate in anaphase, right? Whether we're talking about mitosis or meiosis 1 or meiosis 2, we're talking about separating chromosomes. In meiosis 1, we're talking about homologous chromosomes separating, and mitosis or meiosis 2, and then mitosis, we're talking about sister chromatids separating. But either way, if they don't separate properly, it's called non-disjunction. And that's basically what you need to know for the exam. I'm not even going to ask, again, this part where, yes, technically it happens at anaphase, and I'm not going to ask you that. I'm going to assume you already know that. There'll be a separate question about anaphase. So again, non-disjunction, when the chromosomes do not separate. And of course, when this happens, that produces a gamete that has abnormal number of chromosomes. Think about it. If they don't separate properly, then some of your sperm or egg are going to have too many chromosomes, and some of them are not going to have enough chromosomes. So it'll be very easy to visualize once I show you on the next slide. But yeah, again, non-disjunction. Chromosomes don't separate. If it happened in meiosis 1 or meiosis 2, and I'm going to show you both of those combinations on the next slide. So any questions about this slide before we look at a visualization of these words? Okay, here we go. So on the left, we have non-disjunction in meiosis 1, which is where the homologous chromosomes fail to separate. And on the right, we have non-disjunction in meiosis 2, where the homologous chromosomes did separate, but the sister chromatids fail to separate. Of course, you can see here on the left, if we have non-disjunction in meiosis 1, where the homologous chromosomes fail to separate, then you're going to have all kinds of messed up gametes, right? This one has an extra chromosome. This one has an extra chromosome. This one doesn't have enough chromosomes. This one doesn't have enough chromosomes. In meiosis 2, if the sister chromatids fail to separate, it's a little bit less severe, if you will, I guess, because it separated properly the first time, and you only one of them is failing to separate this time. At least you have one cell that went on to do normal things, right? So you have two normal gametes, and then you have two abnormal gametes. This one, which has too many chromosomes, and this one that has not enough. So any questions about this? I'm not going to get this specific on the exam. It's not like I'm going to ask any questions where I'm asking you to remember the outcome of non-disjunction in meiosis 1 versus non-disjunction in meiosis 2. I'm not going to get that specific. Again, for the exam, you just need to know what non-disjunction is, and that means the chromosomes fail to separate. Whether we're talking about homologous chromosomes or sister chromatids, they fail to separate, and that's what non-disjunction is. Any questions about this slide? All right. The next slide shows an abnormal gamete fertilizing a normal gamete. So again, non-disjunction occurred for one of the sexual partners, right? Maybe the I don't remember which one, we'll see here in a second, but either the sperm or the egg is messed up and the other one is normal. And this results in a zygote that basically has an extra chromosome. Of course, this is just an example, so I'm going to put a line through this because it's not always a zygote that has one extra chromosome, right? There's so many different possibilities. But the point here is, if you have a messed up gamete, right, a sperm or egg, it's messed up, And then when they fertilize, even a regular sperm or egg, right? So you have one messed up, one that's normal. You now have a zygote that's not normal for whatever way, maybe 2n plus 1, maybe 2n minus 1, whatever. The point is it has an abnormal number of chromosomes. But that's how we all started, right? At one point in our life, we were one cell. We were a zygote. And the whole reason we're here is because we went through mitosis, right? You're not one gigantic cell. You're trillions of cells. And that's how it happened. You started as 1, then you went to 2, then 4, then 8, right? You kept doubling. But mitosis, as we know, is basically a process that does a perfect copy of the cell. So if the first cell is messed up and has the wrong number of chromosomes, then every cell after that is going to have the wrong number of chromosomes because mitosis just copies what's there. So if the organism happens to survive, which is not always the case, because like I said earlier, it could be fatal, then they will have an abnormal karyotype, which is another way of saying they'll have an abnormal number of chromosomes, and a syndrome of disorders caused by the abnormal number of genes. So this is, again, could be fatal. Very bad for animals, most animals. Different for plants, if you want to look into that. We call it polyploidy. So in plants, sometimes having extra chromosomes is a good thing, but not very often with animals. And again, you can look that up for independent work if you want. Not just plants. What about fungi? What about protists? Of course, bacteria won't work because they only have the one chromosome. Anyway, any questions about this slide? Oops. All right, let's see a picture of what we're talking about here. There you go. There you have an abnormal egg that has an extra chromosome fertilizing with a normal sperm cell. And then there you go. You have that zygote that has that one extra chromosome. And then, of course, it's going to go through mitosis, and it'll get a perfect copy of that. So that whole organism is going to have an abnormal number of chromosomes. So does anybody know the age when this is more likely to happen for women? Yep, that's what I think it is too. Sometimes it changes, but yes, 35 I think is the correct answer. And if you want to look that up for independent work, you could. Like, why? What is it about being older that makes this more likely for women? And what about men? Why don't their sperm just drastically change when they hit 35? Or do they? And look that all up for independent work. All right, let's talk about what happens when you have an extra chromosome. In this particular case, we were talking about chromosome number 21. And this condition is called trisomy 21, and that's in the name, right? You have 3 chromosomes, because remember, you're only supposed to have 2 of each. But if you have an extra chromosome number 21, that is called trisomy 21. So instead of having 46 chromosomes total, or 23 pairs of chromosomes, right, you have a total of 47 chromosomes and 1 extra chromosome. And anybody who has this has what's called Down syndrome. I'm pretty sure you're probably already familiar with this. I'm not going to ask you these details. I'm going to talk about them very quickly, but I'm not going to ask you to memorize all these things about Down syndrome. But it affects about 1 out of every 850 children. It is the most common chromosome number abnormality, which is why we're talking about it. But this is also a great springboard for independent work topics. So what about the other ones, right? So why is it, what happens if you have an extra chromosome number 22 or an extra chromosome number 17 or whichever numbers you want to talk about, right? Of the 23 pairs of chromosomes, what happens if you have an extra one of the other ones? Or what happens if you don't have enough? The possibilities are endless. Well, not really endless, but there's a lot of possibilities of things to write about. It is the most common serious birth defect in the United States. Like we mentioned earlier, the risk increases with the age of the mother, and 35 years is the cutoff, so to speak. And again, you can look that up for independent work as well. Why? What is it about the age of 35? And why not the father? What's going on with the sperm? Anyway, any questions so far about trisomy 21? All right. Again, I'm not going to ask you these details, but I teach out of the book, so here we are talking about it very quickly. People with Down syndrome have characteristic facial features, right? So usually you can tell if somebody has Down syndrome by looking at them. Very often they have a shorter lifespan. I put a star next to that because it's not always true, but very often it is. There are varying degrees of developmental delays, which is another good topic for independent work. Like why? You know, if you have two people with Down syndrome, why would one be more developmentally delayed than the other? Because it's still just that one extra chromosome number 21. So what makes the difference? Same thing here too with the lifespan, right? Yes, technically you have a shorter lifespan. Some may live to middle age or beyond. Many of them are socially adept. This is again what your book is saying, and you could look this up for independent work. Why? What makes it one person with Down syndrome different than the other? Anyway, any questions about this slide, which is academically unimportant, because again, I'm not going to ask you these questions on the exam. All right, here's a picture of a normal karyotype, right? So this is somebody who has the regular number of chromosomes. They have one pair of each of the 23 chromosomes, right? So up to number 22, and then the sexual chromosomes. And then this next one's going to be somebody with trisomy 21. See right there, you can see there's the extra chromosome number 21. I think there is a question about this on the study guide, and it might be on the test. It's about karyotypes, and basically it says, what can you tell, by looking at a karyotype, which is a picture of the chromosomes, what can you diagnose? That's basically how the question goes. So remember, with a karyotype, you can only see the number of chromosomes, right, and the size. So if I were to look at your karyotype, I could tell if you have too many chromosomes or not enough chromosomes. But I can't read your genes, right? So, I can't look at your chromosomes and say, oh, yeah, they've got the gene for blue eyes or the allele for blue eyes, which we'll talk about later, right? So, again, for the test, for the study guide, at the very least, when you get that question, you can only see the number of chromosomes in a karyotype. Anyway, any questions about this slide? All right. Let's talk about abnormal number of sex chromosomes. And to me, this is really fascinating too. Non-disjunction can lead to abnormal numbers of sex chromosomes, obviously, right? Because we just got finished talking about how non-disjunction can cause an abnormal number of chromosome number 21, right? You could have an extra chromosome number 21. That's called trisomy 21. That gives you Down syndrome. So what happens if we have non-disjunction with the sexual chromosomes? It seems to upset the genetic balance less than the unusual numbers of the autosomes, so less than the other 23 pairs. That's another good independent work topic too. Why? What is it about the sex chromosomes that make it less severe? In this next slide I'm going to show you is going to list some of these very common chromosome abnormalities. And I'm not going to ask you questions about them. So if you're taking notes, you can give your hand a break when we get to this next slide. But again, to me, this is a good independent work topic if you want to look into it a little bit more. So there's a whole list of it, right? So at the very top, we have a male with an extra X chromosome. Remember, Y chromosome is what makes a male for humans, right? So in this case, they have an extra female chromosome, if you will. But it's still a male because they have the Y chromosome. They just have an extra X. And it gives you the origins, which I'm not going to ask you about. It gives you how frequent it is in the population, which I'm not going to ask you about. I'm not going to ask you any of this. But again, I teach out of the book, so here it is. And again, this is to me, very interesting. This would be an interesting independent work topic. Because I just told you again at the top, that's called Klinefelter syndrome. So what are some of the symptoms? Well, of course, there's some of them right there. Sterile, undeveloped testes, secondary female characteristics, but you can look farther if you want for independent work. And of course, there's below that XYY. That's an extra Y chromosome as opposed to an extra X chromosome. Then you have a female with an extra X chromosome, so 3 X's instead of 2. And then you have a female that only has 1 X chromosome, right? Didn't even get another sex chromosome. So those are all things you can look into. And I say this every time I do a lecture, and I always fail to look it up. So I haven't verified this. Could be a rumor, and you can look it up for independent work too. But I think there's something called super males, which would be this XYY. So a male that has that extra Y chromosome. And you can see right there that the frequency in the population is 1 in 2,000. So statistically, if we had 2,000 men in this room and we were to look at all their karyotypes, look at all their chromosomes, and statistically one of them should have an extra Y chromosome. That's in the general population. That part I know. The part I don't know, and this could be a rumor, and you could look it up for independent work if you want, is that super male thing. I think I've heard that the frequency in the population is more than that in prisons. So apparently, from what I've heard, if you have that extra Y chromosome, the one that makes you male, you're a little bit more aggressive. So probably more likely to partake in violent crimes, therefore more likely to be in prison. So if you're a criminal justice major, that might be interesting to you. Or an education major, because, if you're dealing with kids, males, females, some of those males may be extra aggressive, and now you might know the genetic reason for that. Anyway, any questions about this? All right. Again, I'm not going to ask you any of these questions on the exam. Let's see. The next word for attendance will be Turner. Since it's up there, Turner is the next word for attendance. Speaking of attendance, you'll notice I didn't post the video from our last lecture before spring breaks because something went wrong. So sorry about that. Sometimes it just doesn't work. So if you missed Friday's lecture, I guess you're just out of luck. I'll try to post. Well, they're already posted. I recommend there's videos from previous semesters. So I'll try to find the proper videos and share that with you. But anyway, if there's no questions about this, we'll move on. The end of every chapter has this, the evolution connection, right? Every chapter talks about evolution at the end. As always, I'm going to skip it. There are questions about this on the study guide, though. Basically, the advantages and disadvantages of sexual reproduction versus asexual reproduction. Yes, I guess I will talk about it very quickly. Basically, for asexual reproduction, sometimes there are advantages to that. So, for example, it eliminates the need to expend energy forming gametes and copulating with a partner. And I know that sounds weird when you think about humans, but, in nature, energy is important. You don't want to spend a lot of energy. Not only that, but, sometimes it's dangerous to mate. Like think about, what are they? The praying mantis. Very often the female will rip the head off of the male after they're done, right? So, not necessarily a good thing in nature. So anyway, yes, it reduces the need for energy. It also reduces genetic variation. So if you are a perfectly adapted organism and you don't need to make changes and your environment's not changing, asexual reproduction is the way to go. And of course, it's the opposite for sexual reproduction. Read about it if you want. There's questions about it on the study guide. And we are done for the day early. So I'll see you in lab or see you for lecture on Wednesday.